So now it's time for questions, and there are already several questions in the chat. So I'll start with Professor Jiro. And there is a question: what happens to the microbiome of a Japanese person moving, for example, to Thailand or to Vietnam and changing the diet? And does age matter?
Yeah, actually, I don't have a result for such kind of people. Yeah, but actually, we have a opposite type of study result. Yeah, actually, the Thai people who eat a lot of Japanese meal, those microbiota become similar to Japanese microbiota. So that's all what we have. So yeah.
Great, thank you. For Jack, can you comment on the Earth Microbiome Project and how can it help preservation of life microbes from environments and humans in collections?
Is that from you, Maria Gloria?
No.
Okay, so the Earth Microbiome Project is a continuing effort to catalog microbial ecosystems around the planet. And so it can be used, the current database structure can be used to target environments, niche structures, samples that may be useful in preserving functional and phylogenetic microbial diversity. So it can help with the identification of things that need to be preserved. I think that's the best strategy for the how the EMP can be useful.
Wonderful, thank you, Jack. For Professor Zheng, regarding dairy consumption, what about the influence of lactase non-persistence trait? Did you consider this as well? Did you observe any influence of this characteristic?
Thank you, Mario. So I think because in terms of genetic mutation, majority of Chinese people are lactose intolerant, I think. So so I think in our FFQ, we found that overall the dairy intake in our populations, in our cohort, is very low. So somehow I think this low and also for many people the dairy intake is zero. So this kind of trait could be reflected in the FFQ data. So we compare the higher extreme quartile with those lowest quartile or quintile levels in different sensitivity analysis. So I think we kind of already consider this could reflect this phenotype. Thanks.
Wonderful. For Jiro, there is a question. When you sample the populations, are most of the samples coming from urban population? Do you also consider physical activity that has been reported to influence the gut microbe composition?
Yeah, yeah. Actually, we collected the sample both from urban and rural populations, and to compare. So, and what we found so far mainly is the difference in the enterotype. For example, in the Thai population, the sample collected from Bangkok harbored Bacteroides enterotype, while the Thai living in the rural side mainly harbored a Prepotera type. So, and about the physical activity, what we haven't examined the effect of the physical activity on microbiome so far, but at least we have one study maybe related to this physical activity. So we collected the fecal sample from the people living in Bangkok, and one group working in the factory, and the other group is office worker, and yeah, they have distinct microbiome future. So that may be affected by the physical activity.
Thank you. For Jack, based on your study comparing Ghanaian and US participants, can it be assumed that having a very strict healthy diet for lean US participants is necessary to maintain their health due to their unstable gut microecology?
It's complicated, but what we would extrapolate from animal studies and long-term gut stability studies is that it would take approximately 9 months on a very strict diet to remove some of the membership that could have destabilizing potential and to create a stable, robust microbial phenotype, in ecological phenotype in the gut. So I think it does take a long time. The best is obviously intergenerational stability, a reduction in early life antibiotic exposure, a reduction in the presence of foods that are pervasive in our developed societies. So, but you know, I think you could probably get away with 9 to 10 months, and then you would be stable. But you know, pervasive insults to the microbiome and the body will result in a return to that prior state. So the best thing to do is just to eat healthy all the time.
Thank you. For Ju-Sheng, the diet diversity effect that you observe of increasing diversity, what do you think is the mechanism? Is it gaining new bacteria, new diversity, or is just increasing the evenness in the populations?
So I think it's I think it both. It increases overall diversity, evenness, and also affects some beneficial bacterial abundance in our gut environment. And also we found that our gut microbiome derived metabolites are systematically changed in the high diversity group compared to the low group. Yeah.
Thank you. For Jiro, may I know if you consider the stages of the disease suffered by the patients during the sample collection for the study of type 1 diabetes?
Yes. Yeah. In Indonesian study, which I showed today, we haven't collect the stage data of the type 2 diabetes patient. We only collect the information of the fasting blood glucose level and HbA1c. And the patient who showed the pretty strong bacteroides type, which I showed today in the PCA plot, they are very high level of fasting blood glucose level and HbA1c. So we only found that kind of the strong the shift, enterotype shift, only found in the T2D patient, very heavy type 2 diabetes patients, such as the fasting blood glucose level is 300 or so on. Yeah, and now we conducting the more precise study to look into the difference of the stage on the microbiome. So by using a Japanese type 2 diabetes patient, now it's ongoing.
Another question for you is, in risk assessment for NCDs in Mongolian populations, did you find differences, I guess, in urban and rural people's gut microbiome?
Sorry, could you repeat the question again?
The question is, in risk assessment of NCDs, I guess is non-communicable diseases,
Okay.
in Mongolian population, did you see differences in urban versus rural people's microbiome?
Yeah, actually, yeah, as I showed today in the PCA plot, yeah, it is a pretty characteristic that the gut microbiome of type 2 diabetes living in Ulaanbaatar, that is the capital city in an industrialized city in Mongolia, only have the bacteroides type. So, and the urban population doesn't have a bacteroides type. So, yeah, that is only which we have. So,
this is a question for the three of you. Does the microbial diversity origin is from our gut itself, or it comes from the food sources that we eat? Does the location and soil where we grow those food have an effect on microbial diversity in the gut?
From all of our work, I'll start. Sorry, guys. From all of our work, it suggests that no, it doesn't. We do not think there is significant acquisition of microbial components from soil and plants into the gastrointestinal environment. The vast majority of the microbial composition of the gut appears to be acquired from your parents and physical communication you have with human beings in the first few years of your life, probably the first few months, in fact. So that doesn't appear to transition. But what does transition and what does impact the gut microbiome is the chemical composition and diversity of plants that you consume. So, for example, from the American Gut Project, we know that people that consume more than 30 different species of plant a week will have a higher microbial diversity, a higher functional diversity, and a higher stability in the microbial community.
You and Jiro, do you have anything else to add?
I think it's really our human needs those microbiome in our gut, and the diet is only help to shape in that good direction, because we also did a lot of genetic study, because we, for example, in our GWAS for gut bacteria and the GWAS for reason GWAS for gut fungi, and we did find some really actually a lot of genetic determinants for, especially for gut fungi, which is a very minor component of our gut ecosystem. So I do think that our human genetic and physiology need those and also help shape those microbiome in our gut, and the diet of course we help co-shape those microbiome environments.
Yeah. Jiro, you want to add anything else?
Yes, I think it could be the case. So the soil and other environments can be a source of the microorganism for the gut microbiota. Because we don't have the data of the soil, but we have a data of the seaweed. So and there is the fabrication of the seaweed degrading enzyme encoded in the gut microbiome comes from the marine bacteria. So for the vegetable also could be the case.
Right, but in reality, it's not the seaweed bacteria that engrafts in the gut, right? There has been transfer, gene transfer, but not the seaweed bacteria does not engraft in the gut, correct?
I don't know, but of course, in the short period that it doesn't happen, but for the long period for the Japanese history, we have been consuming the seaweed for more than the
in some
generations
hundred thousands of years.
Yeah.
So another question is the timing of feeding. Does it influence the microbiota? Someone mentions that intermittent fasting affects the diversity of the microbiota. Can you comment on that timing and then the periods of fasting?
Yeah, so when you eat during the day can impact which microbes will grow and what metabolites can be synthesized by that community. So consuming more calories and different macromolecular composition of food at breakfast or at lunch or at dinner or in the middle of the night will influence the dynamic stability and functional traits and therefore the host microbe interaction of the population. But then also, yeah, differential fasting has been demonstrated to correlate with changes in the microbiome, although there are no studies that I'm aware that have definitively demonstrated that the changes in the microbiome associated with fasting have any significant shifts, impacts upon human health. That there's some work in mice, but it's limited as far as I can remember.
Wonderful. So this has been a great session. There are a few more questions, but we don't have time. I have a very last question because it's very important that in every country, especially through the scientists that work on the microbiome, there is an effort for preservation. And part of this symposium is done with the purpose of stimulating the creation of local collections to preserve, just to, you can study, but if you collect enough, you can preserve, and eventually you could request a backup of that collection in the microbiota vault. But most importantly of all is that there are local collections preserving the microbial biodiversity. Are there efforts in this direction in China and Japan that you know?
I didn't aware of this specific purpose, project in China. But in our, for example, in our cohort, we did, try to establish a biobank for the collected fecal microbiota and extract those bacteria and preserve that. Just started from last year. Yeah.
Wonderful. What about in Japan, Jiro?
Yeah. Yeah, actually, I don't know in Japan. Yeah, yeah, but some people now stock the fecal sample for FMT application or so on. Yeah, but
So
in the national project, yeah, we have a project to isolate the gut bacteria from the Japanese resources. So
right. I mean, the idea is because we are not very good yet at cultivating the whole diversity, the idea is to preserve the stool sample or the soil sample or the water sample and freeze it until we know better how to culture or how to restore. Especially studies like yours that go beyond your borders, and you have specimens from diverse places in the world. We should preserve that biodiversity before urbanization degrades it for the future. And in that note, I want to thank the wonderful 3 speakers we had today. Thank you so much for participating, helping educate the world in these symposia that are seen by quite a bit of people. There were at some point 80 people this morning, sorry, tonight here. Thank you so much. Now we go for a break before we start the next session, which will be session 2, local infrastructure and research. Thank you so much, and good night and good morning, everybody.
Thank you, Gloria. Bye.